This video will show you how to prepare an agarose gel and perform gel electrophoresis. Gel electrophoresis is a technique that allows you to separate DNA fragments according to size. First, prepare a gel casting tray. Also prepare a piece of masking tape that is longer than the width of the tray. Tape the two ends of the gel tray such that the tray can contain molten agar without any leakage. Make sure that the masking tape sticks firmly to the tray to prevent gel leakage. To be prepared for gel casting, get ready the gel tray, a 50 ml Falcon tube, cyber-safe DNA staining dye, and a micropipette set to 5 microliters with pipette tip attached. Laying out these items on your working space beforehand will allow you to work promptly with the molten agar afterwards. Fit the well comb onto one end of the gel casting tray. To start preparing the agarose gel, place a weighing board on the electronic balance and press the tare button. Weigh out the required amount of agarose powder. In this example, we will prepare 1.2% agarose gel, which is made from mixing 1.2 grams of agarose powder and 100 milliliters of 1 times TAE buffer. Next, add the agarose powder into a clean glass bottle. Next, add 100 ml of 1 times TAE buffer into the glass bottle. Swirl the bottle to mix the contents well. Loosen the bottle cap and place the bottle into the microwave oven. Heat the liquid for about 2 minutes to boil. Put on oven gloves and check whether the agar powder is fully dissolved. The molten agar is a thick liquid and should appear clear like water. If you see powder residue or if the liquid is not clear, heat the liquid again. Once the molten agar is ready, pour 50 ml of agar into the Falcon tube. Next, add 5 microliters of cyber-safe DNA staining dye to the molten agar. Replace the cap of the Falcon tube and turn the tube upside down a few times to ensure that the dye mixes well with the molten agar. You should be wearing the oven gloves as the molten agar is still very hot. Pour the molten agar into the gel tray in a steady stream to prevent bubble formation. If you see bubbles on the agar, use a clean pipette tip to remove the bubbles. Leave the agar for about 15 minutes to solidify. After the agar has solidified, remove the well comb. Carefully remove the masking tapes from the gel tray. You should see well-defined rectangular wells in the gel. Next, prepare the gel tank and place the gel tray into the tank. The wells should be closer to the cathode or the black end of the gel tank. Since DNA is negatively charged, it will migrate towards the red end or the anode of the gel tank. Next, pour 1 times TAE buffer into the tank until the agarose gel is fully submerged in the buffer. Next, use a micropipette to load 5 microliters of DNA ladder into the gel. In this example, the 100 base pairs and 1000 base pairs ladders are loaded into the gel. DNA ladders contain fragments of DNA with known sizes. They serve as size standards so that you can determine the size of your DNA sample analyzed in the same gel. To start loading your DNA sample, prepare a strip of parafilm and pipette 5 microliters of your DNA sample onto the parafilm. Next, add 2 microliters of DNA loading dye to the drop of DNA sample. The color of the dye helps with sample loading and allows you to see how far your sample has migrated. Next, use the micropipette to draw up all 7 microliters of sample and load it into the gel. Mix the DNA sample with loading dye and load the DNA into separate wells for all other samples that you wish to analyze. You may also use PCR tubes instead of parafilm to mix your samples with the loading dye. Make sure that you use a new pipette tip when you are handling different DNA samples. When loading the samples, the pipette tip should be immersed in the buffer solution and positioned just above the well. Do not lower the pipette tip to the bottom of the well. You should dispense your sample slowly and steadily. You should record down the well number and the identity of the sample loaded into each well. After loading your DNA samples, you should see small dark blue rectangles in the gel. Place the gel tank cover onto the tank and make sure that it fits firmly. The red cable should be at the red end of the gel tank and the black cable at the black end. Connect the cables to the power source, keeping in mind the rule red to red, black to black. Next, turn on the power pack. The light below the letter V or voltage will be lit. Press the up arrow to set the desired voltage. Press the display button twice. Press the up arrow to set the desired duration of the gel run. In this example, the duration is set as 60 minutes. Press the Run Stop button. You should see tiny bubbles emerging from the cathode. During the gel run, you should see the dye front migrating down the gel. When the gel run is completed, turn off the power pack and unplug the cables. Next, open the gel tank. Carefully pick up the gel tray with the gel and place it on a plastic tray. Bring the gel to the gel documentation system. Carefully slide the piece of gel onto the surface of the gel dock. Do this slowly as the gel is very fragile. Open the Gene Snap program on the computer. Select the upper white option to turn on the white light and click the green button to view a live image of the gel. You may put on gloves to adjust the position of the gel. Remember to close the chamber door once the gel is in position. Select the Trans Illumination option. With UV illumination, you will see DNA bands on your gel. Click the red button to capture the image. To save your image in a format that can be analyzed by the program, click File and then Save As. This will save your image in the SGD format. To save your image in the TIFF format, click File, Export Image to TIFF. This will save your image as a TIFF file. Remove your gel after the imaging is done. Use Kim wipes to wipe down the surface of the gel dock. Place the unwanted gel on paper towels and leave it to dry. The gel will shrink and dry out within 2 days. Finally, discard the gel into a biohazard bag.